Welcome, students, to the second part of this chapter. And actually, to understand kind of the next section as we talk about how muscles function, we actually have to kind of backtrack a little bit because we have to talk about resting potentials in all of our cells in our body, as well as the difference between graded potentials and action potentials. So we're actually going to take a little bit of information from chapter 3 and a little bit of information from chapter 11 so that we can then discuss how excitability works in our muscle tissues. But before we can talk about excitable cells, we have to talk about all of our body's cells and how they are polarized, meaning they have a voltage across their membranes. It's approximately 70 millivolts in all of our cells, and all of our cells have a resting membrane potential. They have this kind of voltage difference, this current across the membranes, where the inside side of the membrane is negative compared to the outside side of the membrane. What makes this resting membrane potential is the fact that we have different ion concentrations inside our cells and outside our cells. The ions that we happen to be talking about are sodium and potassium. They're the ones that are mostly responsible for this resting potential. In terms of sodium, we have more sodium in the extracellular fluid than we have in the intracellular fluid. So more sodium outside the cell than in. For potassium, we have more sodium in the intracellular fluid than in the extracellular fluid. So more potassium inside the cell than we have out of the cell. So we have different ion concentrations. Well, when ion concentrations are different across a membrane, if you have a high concentration in one area and low concentration of that same ion in another area, they're going to diffuse. But in terms of our cells, they don't diffuse evenly. The plasma membrane is much more permeable to potassium than it is to sodium. We have more ion channels for potassium than we do for sodium. The net result of all of this, as both of these ions kind of diffuse, more potassium diffuses out of the cell down its concentration gradient than diffuses into the cell down its concentration gradient. Now, those diffusions would continue until both sodium and potassium were in an equilibrium across the membrane. Unfortunately, if that ever happened, that would kill the voltage difference and we would have no more resting membrane potential. And if we have no resting membrane potential, then we don't have any way to make action potentials and have our nervous system function or our muscles function. But fortunately for us, that is not what occurs. Sodium never reaches an equilibrium across the membrane. Potassium never reaches an equilibrium across the membrane. And that is because we have the sodium-potassium pump, the sodium-potassium ATPase, that essentially maintains those concentration gradients. Sodium diffuses in, but the sodium-potassium pump pumps it out. Potassium diffuses out, but the sodium-potassium pumps it against its gradient back into the cell. That keeps the high extracellular sodium and the high intracellular potassium. And by maintaining those concentration gradients, the resting membrane potential is able to be maintained. Let me show you a diagram of what I'm talking about, and then we're going to watch a little video. So we have a higher sodium concentration outside the cell and a higher potassium concentration inside the cell. The potassium diffuses out, the sodium diffuses in, just down their concentration gradients, just leaky ion channels that are open all the time, allowing these two ions to move. But you'll see we have many more potassium channels than we do sodium channels. So some sodium diffuses in while some potassium diffuses out. But since we have more channels for potassium, we lose more potassium than we gain in positively charged sodium. And so that results in this kind of negative potential on the inside of the membrane versus the outside of the membrane. let's talk about the sodium-potassium pump. That's what you're seeing here, right? So the sodium diffuses in, potassium diffuses out, because they have concentration gradients that they can diffuse down. If we did not have the sodium-potassium pump, sodium would diffuse in and potassium would diffuse out until both sodium and potassium were in an equilibrium across the membrane, meaning equal concentrations of sodium in and out, equal concentrations of potassium in and out. That would kill our resting membrane potential and would also kill our ability to send action potentials and basically kill the functioning of our nerves and our muscles. But the sodium-potassium pump pumps 3 sodium out for every 2 sodium in, so that keeps the high intracellular potassium concentration and the high extracellular sodium concentration. If that's still confusing to you, we're going to watch a little video because I like to see stuff move. Now, this particular video is going to talk about nervous cells in particular, but resting membrane potential actually applies to all of the cells of the body.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
So to reiterate, right, we have the sodium and potassium pumps pumping sodium against its concentration gradient out of the cell, and potassium against its concentration gradient into the cell. That allows potassium to leak out and sodium to leak in down their concentration gradients. And that is what sets up this interior -70 millivolts versus a positively charged exterior portion of the membrane. That's what sets up this resting membrane potential. Again, the video showed you specifically a neuron, but resting membrane potential applies to all of our body's cells. But muscles and nerve cells are different. Muscle cells and neurons, nervous system cells, they are excitable. And by excitable, we mean that they can change their membrane potential. They don't always have to be in a resting state. They can change it. They can become excited. And they change their membrane potential by changing the ion concentrations across the membrane. To change the ion concentrations across the membrane, you have to change the permeability of that membrane to these different ions. So we're going to open and close some gated channels and give these ions essentially more doors that they can move through. And when they move through these different doors, either into or out of the cell, that then changes the ion concentrations across the membrane and thus changes the membrane potential away from that resting value. So if our resting value is negative 70 millivolts, there are essentially two ways that we can change that, either up or down, as we'll get to in just a second. The main ions that we generally talk about in all of these scenarios is sodium and potassium. Sometimes calcium is involved, sometimes chlorine is involved, but primarily we're talking about sodium and potassium. The resting membrane potential is using those leaky channels, sodium out, potassium in, to kind of set up that voltage difference. But these changes in these membrane potentials, either up or down, have to use a gated channel, right? It's going to open or close. And there are two ways that gated channels open or close. You can have a chemically gated channel when some ligand, some chemical messenger, binds onto the channel opens up. This is the kind that we find in our synapses. And then you can have voltage-gated ion channels when they open or close depending on the membrane potential. If the membrane potential changes, that will trigger these kind of voltage-gated channels to open. So, right, we're going up or down, right? And we're going up or down in terms of changing our membrane potential in relation to that resting potential value. If we make the membrane less negative, closer to zero, even potentially positive, then that is called depolarization. It's actually a decrease in the membrane potential, right? Meaning the polarity, the difference between the inside and the outside is getting less. So depolarization is when we make the membrane potential kind of less negative, potentially positive, which is usually due to the influx of some positively charged ion like like sodium, but sometimes it's calcium. If we hyperpolarize our cell, right? Hyperpolarize means more polarized. So we're going to increase the difference between the inside and the outside of the cell. To increase the difference between the outside or inside of the cell, then the membrane potential is going to increase, right? We're going to get further and further away from zero, meaning the inside of our cell is going to become even more negative than the outside, so that potential difference is larger. Typically, this is due to the outward flow of some positively charged ion like potassium, although sometimes it is due to the inward flow of a negatively charged ion like chlorine. But these are two terms that you should know: depolarization and hyperpolarization. Depolarization, the inside becoming less negative; hyperpolarization, inside becoming more negative. And in both instances, when we return to our resting value, that is referred to as a repolarization. So we can depolarize and repolarize, we can hyperpolarize and repolarize. When we change these membrane potentials, two types of signals are possible in our body: what we call graded potentials and what we call action potentials. Graded potentials are short distance, kind of local signals, and they will depend on the strength of the stimulus. They're tempered, meaning, right, you have a grade, right? An A is different than a B, 95 is different, 95 degrees Fahrenheit is different than 80 degrees Fahrenheit. There's a grade, they're a gradient. Action potentials are all or nothing. They are roughly the same change in membrane potential. the same size, kind of no matter how strong the stimulus is. They either happen or they don't. That's what we refer to when we talk about all or nothing. And these are long distance signals. These are not just used locally, but they are used for your brain to communicate out to your toes so you can wiggle your toes. But using these different potentials, graded potentials, action potentials, is how our body cells send and receive and integrate information. So let's talk about the graded potentials first. Again, these are short-lived, localized changes. Only the membrane in a very small portion of the cell, or maybe potentially a couple of nearby cells, is going to change. Now, graded potentials can be either depolarizing or hyperpolarizing. So sometimes a graded potential is going to bring us closer to zero. Sometimes a graded potential is going to bring us further away from zero. And they are graded. If you have a stronger stimulus, you get more voltage-gated channels opening. You get a bigger graded potential that can then travel a further distance. The problem with the graded potentials is these flows are dependent on these opening of these ion channels, and they can't really, they dissipate. They think about the further away you get from like a stream of water or stream of air, right? It feels, if you stand right in front of the air conditioning unit, you get a lot of air. But if you step further back from that, you don't get as much air. And that's essentially what happens with these graded potentials. They're really, really good over short distances, but over long distances, the strength of the signal kind of decays. I'll show you what I mean. So here we have a small portion of the membrane that has been depolarized. That's what you're seeing here in red. It's becoming slightly positive inside the cell as opposed to negative. And that's going to affect ion channels in the nearby kind of parts of the membrane. So we get all these little local currents, these little local membrane potentials that are changing, and that kind of spreads the depolarization. And you can see here's this kind of active initial depolarization, and the areas near it are pretty strong, but the further we get away from where the stimulus was applied, we're getting less and less kind of changes in the membrane potential, kind of less and less signal. So graded potentials, short distance only. They can only really affect kind of just that local area. Action potentials are different. Action potentials are able to travel long distances. They do not decay like graded potentials do. Action potentials are always depolarizing. It is always a roughly 100 millivolt change from negative 70 up to about positive 30 in terms of the inside of the cell actually becoming positively charged. And action potentials is what is occurring in muscle cells and in our neural cells, particularly the axon part of the neuron, as we'll discuss when we get to Chapter 11. But it is these action potentials and their ability to kind of travel long distances that allow neural communication between muscle cells and neural cells, that are really responsible for what makes muscle cells and neural cells really just being amazingly different and special and compared to some of the other body cells. Not that those aren't, unique and needed too. And that is the end of that. We will talk more about action potentials and graded potentials when we actually get to Chapter 11. But to understand how muscles function, you do have to have at least the basics of depolarization and hyperpolarization and resting membrane potential, which is why we covered it here.